So this is actually, I've been called many different things in my life. I've been called a scientist, I've been called a geneticist, I've been called a hacker, I've been called a futurist. Inventor is kind of a new term. What I've learned in all of these different roles is that if you see things that other people don't, they generally think you're crazy. This is a polar bear in a snowstorm. Now, it turns out because I study very small things, and because I study information, most of the stuff that I think about is in fact invisible to most people. Really, whether it's the internet, whether it's microbes, whether it's DNA, or thinking about the future, these are things most people just really can't visualize very easily. Now, I can't really see the future, but I can extrapolate a lot of different trends. And the past is a really good guide for sometimes how things move forward. Because I sit on the intersection of biology and computing, I draw a lot of comparisons between these two different areas. Now, computers, we all know. We grew up with them. We've built all these systems. We understand the components, the circuits, the systems, the computers, and the networks. It's all very intuitive, particularly to young people. What isn't so intuitive is biology actually created these same architectures over the 3.5 billion years of evolution, starting with very simple components, making more sophisticated circuits, ultimately making metabolism, making cellular processes, organs, organisms, and ecosystems. I find this really fascinating. And there's a lot of similarities between these architectures, but there's differences too. And the key ones is that all computers are programmed in binary code, zeros and ones. And of course, these living systems are programmed in genetic code, A's, T's, G's, and C's. Also, probably a little less intuitively, these living systems, even a single cell, are just so much more complex than any of the computing systems we've ever built. We can't model the simplest cell in the most powerful computer today. When it comes to working with these systems, though, if you just learn one language, DNA, you can actually reach into every single living organism. You can't say this for computers, where there are hundreds of different languages, always changing, always shifting. So DNA is actually the most powerful language, a programming language on the planet. Makes it a very powerful language to learn. And I did quite early on. And I started to work for a major biopharma corporation. This company literally rewrote DNA, or wrote DNA to reprogram cells to make valuable products, in this case, cancer medicines. And it was an amazing experience. We helped a lot of people. We grew very quickly. We were profitable. It was really a dream job. But the longer I worked with them, the more uneasy I felt. And this was really palpable after the 7 years that I spent with them. And I couldn't really put my finger on it until towards the end. And I realized it's because on one hand, I'm working with these electronic systems, which, as you know, get faster, better, cheaper all the time. They're always changing. They're evolving very quickly because of Moore's law. Moore's law is incredibly, it's a part of our modern world. We expect it. The whole industry is driven by Moore's law. On the other hand, drug development is not exponential. This is a graph of the efficiency of drug development output in terms of new drugs per $1 billion invested over 60 years. And it's a negative slope. What this means is you get fewer drugs per dollar invested over time. And it's been that way since we started really measuring it. This isn't a problem with one company or one country, IP rules, et cetera. This is a fundamental flaw in an industry, one that isn't going to change over time. It means that pharma's bones are essentially rotting and will fail at some point in the future. And practically, it means that fewer people are going to be able to afford life-saving medicines because they're getting more expensive to make. This is not Moore's law. And in fact, you can see this is an exponential decrease. This is an example of what we call Eroom's law, the reverse of Moore's. And it's a fatal flaw. I really didn't know what to do. This deeply troubled me after a while. But a couple of bright points popped up. One of them was one of my employees, a programmer, introduced me to the idea of open source software, specifically Linux. Now, Linux is an open source operating system that's grown since the early 1990s. And when I first got introduced to this, it was really quite remarkable because it was actually, even though it was written by an army of volunteer programmers, it was successfully competing with Microsoft in specific areas, primarily servers. This really opened my mind to the power of communities. I also went to this little place. If you don't recognize it, this is a satellite image of Burning Man. It's a temporary city that pops up in the middle of the Black Rock Desert in Nevada, about 2 hours from Reno. It's ephemeral. It comes in at the end of August for about a week. It's an open city. It's a vibrant city. It is an absolutely incredible place to go and explore. And yet there's almost no supervision or regulation. This was a really new idea for me. And just as quickly as it appears, they have a big party, they burn the man, they burn a lot of other installations, and then everything goes away, leaves no trace. These were ethoses that I'd just never come across in society before. And it really deeply affected me and made me realize that communities and volunteers and openness could do remarkable things. This truly is one of the best parties in the world. And I came back to my pharmaceutical job and I realized, I gotta change my life. I gotta think differently about things. I gotta do things differently. So I rebooted everything, sold everything off, put the few things that I couldn't sell, the keepsakes in the storage, and I went to Thailand. And I went to Thailand for no particular reason other than I thought it was beautiful and it was probably inexpensive and it would be a good place to actually stop and think about my next steps. I learned a lot of different lessons in Thailand. I learned how not to be a workaholic and to focus more on play. I learned that intellectual property and copyright really doesn't matter to some countries. It was amazing. A movie would come out anywhere in the world or a CD and you'd have it the next day. And I learned a lot about drug development because there were some remarkable parties in Thailand. And I have to say, the lesson there was, you know, we think it costs $1 billion in 15 or 20 years to make a new drug. No! It only takes that long and costs that much if you're trying to make a legal drug. And trust me, the clinical trials here were done on a massive scale. So I took these lessons, and I went back and I started thinking about something I was a little more familiar with, cancer. Because, of course, cancer is what really powered all of my genetic explorations. It was the funding mechanism. And I had a really different idea about cancer than most people. I just looked at it as a corrupted hard drive, not as a disease. Like, corruption happens. It didn't really matter why a disk corrupted. What really matters is that you can identify the corrupted disk and either delete it or reformat it. Now, by this time, the cancer genomes had started to be sequenced, and we knew definitively that no two cancers were the same. So the whole idea of a mass market cancer drug, or the old school methods of carpet bombing cancer, that wasn't going to work. We really needed personalized medicines. Unfortunately, and I knew this from my time with Pharma, they worked on kind of a blockbuster business model, just like Hollywood. They make a product that will satisfy the most people, really hitting around the mean. And I knew that's not what I wanted for cancer, if I got it. I knew it's not what I wanted for my kids or anyone that I loved. I knew we had to make a new model for a new business model for personalized cancer medicines. Now, there was no model to follow. There was nothing I could use as a template, because this was an industry-wide problem. So I had to start with just a blank sheet of paper. I was out to reinvent drug development, and I thought, I must be crazy. I'm one guy sitting here with a pencil. But I had a starting point. I figured if all the companies weren't solving the problem, why don't I just do the exact opposite what all the other companies do? And all the companies were for profit. So, okay, well, let's make a community-based biotech rather than a corporate biotech. Let's make it open rather than proprietary. Rather than make a mass market drug, let's focus exclusively on making drugs for one person at a time. Mass customization, so to speak. And rather than sell it, let's give it away. And that was just my template for this little crazy company that I made called the Pink Army Cooperative. It is, I learned, the first cooperative biotech company in the world. And it was really a thought experiment, you can imagine. But I had some secret weapons. And one of them is, I was a bioinformatics expert. I used tools like this to design genetic material all the time. These are online tools. This is essentially a genetic editor. And you can do remarkable genetic manipulations with these tools to design DNA. And they can be shared. So I knew we could actually design, as a community, genetic code quite easily. Then there was a technology I'd been tracking for years, DNA printers. These are literally machines that synthesize new DNA. It's like a DNA 3D printer. This is amazing because before we had to manipulate DNA in very manual ways on the bench, cut and splice, glue things together, kind of like making ransom notes, except with molecules. This, you could just print out the DNA, and the machines were fairly powerful. But they weren't so powerful that I could make a large construct. So I didn't know what type of medicine to make. I knew I couldn't make a single protein because that wouldn't be personalized. So I was scratching my head on this, and one day, a friend of mine came into my office and put down a paper on my desk, and he goes, "Read this." And it was a review article on something called an oncolytic virus. Oncolytic means cancer breaking. And it turns out that there are weak viruses in the world around us that are so weak they can't infect a normal cell, but they can infect a cancer cell. And they get inside and they start to replicate and they kill the cancer and they release more virus, which goes on to infect more cancer cells, and you actually can get measurable decreases in cancer because basically cancer cells catch a bad cold. And this was so elegant, and I knew that the DNA synthesizers would allow me to make these types of medicines. So essentially, I started researching synthetic oncolytic viruses, making medicines for one person at a time. And I knew that we could do this so inexpensively that it could really be disruptive to the industry, in part because you no longer had to do large phase clinical trials, the most time-consuming and expensive part of developing a new medicine. But the co-op was, although it was provocative, it was too early to go viral, no pun intended. People couldn't see it. So for the last few years, I've been very fortunate to work with this company, Autodesk. It's a company that does computer-aided design for basically every other product in the world, buildings, cars, etc. There's about 8,000 employees. We have a large suite of software for really designing the world. And my group's job was to bring this experience to biotechnology and nanotechnology. So we built a viral DNA editor. We 3D printed these virus particles. Some people actually thought they were infectious. We pushed the limits of DNA synthesis technologies and actually made whole genomes. And then we demonstrated that it could be booted up. This is actually synthetic viruses killing E. coli. Now, I want to be clear. These are not cancer-fighting viruses. These are antibiotic viruses. but the process is essentially the same to make any virus. What I really didn't appreciate was just how much response this little project would get. It got picked up by the media. Internally in the company, it was mind-altering in some ways. It changed perspectives. And even the Museum of Modern Art acquired all of the assets of this project and brought it into their collection as the first synthetic organism in the MoMA. So what's next? Well, we keep going. The work continues. We're making better tools today for viral design, even data-driven design. We're pushing the limits on simulation and modeling, etc. We're also testing out new desktop DNA assembly systems that are quite powerful now and getting more powerful every day. And we're pushing those into robotic cloud laboratories. So now the entire process of doing this type of drug design is essentially digital. And because digital things get faster, better, cheaper, I'm pretty sure that over the next few years, we're going to make the cost of making a single virus down to a few pennies. I'm looking forward to getting this into canine clinical trials, working with virologists and veterinarians over the next few years. But this is really the start of a completely new drug development pipeline that can affect things like gene therapies and vaccines and so much more. And eventually, we'll move beyond just the viruses, because whole genomes can be synthesized today, bacterial and soon yeast. So the next generation of biotech companies are really going to be people just sitting around with laptops and cell phones and all of this digital infrastructure to support them. So in closing, if you see something that others don't and a path to achieving it, you're not crazy. You could be a futurist, or really you could just be an inventor. Thank you.